So in this track, we're going to review clinical trials for several lupus treatments. We have several compounds that are in late stage testing, but we have some that are in earlier stages of testing. And I'll go over the results and also the mechanisms involved. Here are my faculty disclosures. Now, lupus is complex, but don't panic. I call this Immunology 101. So let's start at the top. In lupus patients, the plasmacytoid dendritic cells are making too much interferon. And this happens in about 50% of our patients. It's called the interferon gene signature. It has consequences. When there's more interferon, there is going to be more activation of the myeloid dendritic cells. And these are important for two reasons. They make BLyS, the B lymphocyte stimulator factor, or BAFF is its other name. And this is what keeps B cells alive. It's sort of a survival factor. But also myeloid dendritic cells present self-antigen to the T cells. And of course, we know in lupus, there's too much T help, there's not enough T regs. And so now the T cells are going to activate the B cells. And the B cells, by making autoantibodies, will end up with the formation of immune complexes that can now activate the plasmacytoid dendritic cells. So we put this circle around, and this circle is a feed-forward loop, so it's going to keep going around. I like to think of this as an equal opportunity slide. There's so many different places where we could break this cycle. And perhaps we need to personalize it. Perhaps in some patients, breaking the cycle at one point is more important or more effective than breaking it at another. So I think someday, before we start any new treatment, we'll have some simple genetic proteomic test that will tell us which biologic or which small oral molecule to pick.